Thank you so much. So tonight's meeting, our February 12th meeting, our special guest speaker is David Tarpy, and he is a professor of applied ecology and in the North Carolina Extension Specialist in honeybees. So he's joining us very late at night. It's literally 9:30 where he is. So we're very grateful that he stayed up late with us. Um, among other extension initiatives, his program runs the Queen and Disease Clinic and the Beekeeper Education and Engagement System, or also known as BEES. His research interests focus on the biology and behavior of honeybee queens in order to better improve the overall health of queens and their colonies. His lab focuses on the reproductive potential of commercially produced queens, testing their genetic diversity and mating success in an effort to improve queen quality. He has served on the boards of the North Carolina State Beekeepers, the Eastern Apiculture Society, the Bee Informed Partnership, and the editorial boards of the top two scientific journals on apiculture. He's a highly sought after speaker. In fact, he's was just at a huge national conference a few weeks ago, and we are very grateful to have him. This is the second time he's spoken to our club and streamed live with us. He's always in high demand, so we're glad he's here. So, with that, I'm going to go ahead and stop sharing my screen. And David, welcome. We're so glad you're here.
Yeah, no, thank you, Sheree. Appreciate the invite back. I had a great time last time, and I'm thrilled that you guys weren't so scared that you invited me again. So it's always great to connect with you guys back in my own stomping ground. I don't know if anybody told you, I got my PhD right up the road in Davis. So I've spent quite a bit of time out there. In fact, that's where I really started thinking about some of the ideas and some of the things that I'm going to be presenting here based on our science. And I think I titled this "Diagnosing Queen Problems." So if you think you're going to get answers or anything, I'm afraid you're going to be really disappointing. Because really, I'm asking the question, is it the queen's fault? Is it the colony's fault? Is it both or is it neither? And the answer to all of the above is yes. But really, the purpose here is to just kind of have a discussion about queen problems in general and kind of following my thinking and my arc of my thinking about this issue and where I stand now, which is really looking at it kind of 180 degrees opposite of how I went into it. So we'll just kind of go through it and have that exploration together.
David, quick question. How would you like to handle questions? Would you like?
Um, Totally
Mhm.
up to you. I think I saw something come in on the chat, but why don't you, because you have the mic there, why don't you
Mhm.
chime in and you're free to interrupt, but feel
Okay.
free to chime in if there's any questions as we go along.
Sounds good. Thank you so much.
Great, thanks. So based on lots of different surveys, queen failure, queen problems, queen issues, these are often in the top five or so reasons of why colonies are dying these days. So this is a pretty common thing that beekeepers are seeing where, 20, 30, 40% of colonies are having queen issues. And this can be really problematic for beekeepers because obviously having a good, strong queen in your colony is paramount to having a good colony. One of the issues that I think beekeepers are at least anecdotally seeing is that queens just aren't living as long as they used to. I think if you talk to beekeepers that have been in the biz for, 20, 30 years, back 20, 30 years ago, even, after Varroa came, queens, you would replace your queen, maybe every 2, 3 years. Commercial beekeepers would, only do some of their colonies every other year kind of thing. And now commercial beekeepers are routinely requeening every single colony every year, if not multiple times a year. People aren't seeing these the queens that live 5 years in the textbook. You don't really see that very much. Problem is, we don't have very good baseline data on how long queens are supposed to live, right? I mean, the textbooks say, 3 to 5 years, but like, we don't have good kind of empirical data. The best I could come up with is I found this paper out of Canada where Tibor Szabo had marked all of his queens, and he noticed every time he went into a colony when the colony had an unmarked queen, so he knew that the old queen hadn't lived as long, and this is what it looks like if you look at the 50 percentile, so the median lifespan of queens in Saskatchewan in the 70s and 80s was about 2 years, right? So 50% of the queens, half of them live longer than that, half of them live shorter than that. We took some data on a study we had done here in North Carolina several years ago, and um, we were finding something completely different where the survival was less than a year of these queens, or the median survival was less. Ernesto Guzman Novoa did a study down in Africanized bees and showed that they even, lived even shorter, So like there's genetics involved, there's, kind of the latitude of where these are taking place. There's lots that go into it. But again, anecdotally, I think a lot of beekeepers are saying, well, we're just not seeing queens live as long as they used to. In fact, we did this study back in the CCD days kind of thing where we were following colonies and commercial operations for an entire year. And, most of them didn't live the whole year. We were tracking those queens. And one thing that we did right in the very beginning is that we sampled the worker offspring from each of these colonies. And then we did these kind of molecular paternity analyses on them. In other words, we're finding out who their fathers are. And queens mate with a whole bunch of males. And so you determine the paternity test on enough workers, you can kind of count up the number of fathers that sired all of the workers, and that's the mating number of the queens. And so what we found was that queens that had mated with a sufficient number, that is above seven, they were 3 times more likely to live than the queens that mated less than that. So, this is an invisible factor. You can't look at a queen and know how well mated she is. You don't can't look at colonies and see how many drone fathers, are in the worker force, how genetically diverse they are. But this has real life consequences. If a queen is just not mated well, the queen and the colony are 3 times more likely to die. That's a very significant, factor. We've also done other studies, looking at pesticides and the cumulative kind of toxicity of the wax within the brood nest. That cumulative toxicity measure is often called the hazard quotient, which adds up all the different pesticides that's in the wax, but it weights them by their toxicity. So, you know, a large amount of something, very benign, like a fungicide or something like that, doesn't add much to that hazard quotient. But even a little bit of something that's really, really toxic can add a lot to it, right? So it just weights it by that. And colonies that were experiencing issues with their queens had significantly kind of higher levels, cumulative levels of pesticides in their beeswax compared to those that don't. So, clearly the nest environment has something to do with how long queens live and whether colonies, decide to replace them. Then again, looking at the effects of pesticides on queens themselves, we had done some studies where we're looking at um, increasing dosages, like 30 times what you actually might see within, a real world environment. We weren't really seeing a lot of these different pesticides affecting the queens, their body weights, and the viability of their stored sperm. And so, I think the issue that pesticides have on queens is more indirect and not direct. It's not actually affecting the queen per se, but it's affecting the colony. And we'll come back to this towards the end. That pesticides and the effects on queens is very complicated. But in the end, if a colony undergoes a swarming event, a supersedure event, some sort of queen problem, those colonies that undergo those events are 3.1 times more likely to die than colonies that do not. And that's because when a colony undergoes a queen event, like a swarm, um, you don't have a laying queen, so there's a pause in the rearing of brood. You don't have the colony growth that you should. And you can often have these cascading stressors that intervene, like mites, like nutrition, like other things. So if you have this major perturbation in the colony with losing its queen, it has real world consequences downstream. And so it's just one of those things that you really, really want to avoid queen problems in your colony to keep them alive. So, so I'm going to back up a little bit and, not talk about kind of the any specific thing, but really go over the litany of different things that can cause good queens to go bad. There's been a lot of study looking at all of these one by one, and how these kind of things affect queen quality. And I'll just kind of, for convenience, put them into different categories, these different buckets, right? So there's kind of biological reasons of the queen herself, like is she infected with diseases? Chemical communication and different pesticides, as I talked about, management issues that we include, like treating for mites and the stocks that we use and everything else. And then environmental factors. What are the bees exposed to in their surrounding environment, and what are they bringing back to the nest, right? All of these things have been looked at when it comes to queens, and some of them more important than others. Um, but I would argue that the term queen problems, queen events, um, that's really a catch-all phrase. And I would submit to you that really there's at least four, probably more, different phenomenon that are going on that we kind of lump together as queen problems. But I think they're very distinct. And so understanding the distinctions are important. And then also talking about different reasons that can cause them, I think really helps to inform how to diagnose queen events in your colony, because not all queen events are the same. Okay. So we're going to talk a little bit about just the poor queen quality of the queen herself, the bad brood pattern that she can be laying, what she is laying, that is it fertilized or not? And then, as kind of I mentioned before, the reduced longevity of queens as a separate issue. Okay. Now, if you go into a colony and you have a queen event and you look at the bottom of your boot and there she is smashed, on the bottom of your shoe, that's a beekeeper induced problem, not an actual kind of biological phenomenon. Um, so, that's not what I'm talking about when I'm talking about bad queens. When I'm talking about bad queens is small, runty, not very good queens, right? Bigger queens are better. And there is actually quite a bit of variation among queens in their body size. And part of that can be the way that they're reared. So, if you allow a colony to just replace its queen and naturally replace its queen, they will do so by taking young worker larvae and start feeding it royal jelly. And then they transform the comb, right, so that it's not horizontal, but it goes vertical. And by doing that, what happens is that that worker or was worker, now being fed royal jelly, develops into a queen. The problem is that the bees sometimes they feed not just newly hatched larvae, but they'll feed 1, 2, or 3 day old larvae royal jelly because they're in such a rush to get a queen as soon as possible, right? Well, what happens is that those larvae that are already spent a day or two developing as a worker, they don't get as much royal jelly. They don't develop into a full-fledged queen. They do develop into queens, but they're more worker-like. So you get these intercasts, you have the queens, which are the biggest and, the best, the workers, which are sterile and, not at all reproductive. But you can get these intercasts depending on what age the larva is when it's initially fed royal jelly. And so walkaway splits and, those types of ways of raising queens, they can often raise their queens from older worker larvae, and so the queens aren't as good. They're not as big, they don't mate as much. And so, you know, kind of you need to beware when you're allowing the colonies to make their own queens. Most of the time they do prefer making queens from the youngest possible larvae, but not always. So kind of beware and don't be surprised if the outcome of that is kind of a smaller queen. Now we're scientists, so we like to test these kind of things. And so what we did is purposely raised queens from older larvae versus younger larvae. So we had high quality queens, the best queens that we could make, and then the worst queens that we could make on purpose, right? So, and then we placed these out into packages and we, installed them into new equipment and then followed them over time. And sure enough, colonies headed by high quality queens, those colonies grew stronger, bigger, better over the course of the season than colonies headed by low quality queens. Um, Now, this difference isn't really night and day. It is statistically significant, but it's not kind of, grotesquely obvious or anything. And so the way I look at it is that if you have a runty queen, a low quality queen, um, the ceiling for her colony is going to be a bit lower, right? And if you have a high quality queen, the ceiling for that colony is going to be higher. But you can take a really awesome queen and put them in a colony, and they are still stressed from pesticides to, poor nutrition, all these other things. The colony is still not going to be very good, right? But all things being equal, if you have a better queen within your colony, it's going to have a higher ceiling for success. Right? Now, that's what we're talking about when we're talking about the quality of the queen herself. But I would submit to you that the vast majority of beekeepers do not look at the queen and say whether or not she's of high quality. They look at the brood pattern. They go into the colony, and if they see something like this, which beekeepers call shot brood, then they say, oh, this is a bad queen, get rid of her, and your problem will go away, right? This is taught in the textbooks. This is, just kind of a knee-jerk reaction by most beekeepers. And there is truth to this, that if you see shot brood, it can be a problem with the queen. And that stems from her mating and actually a mismatch of mating with the genetics. So not to get too deep into like honeybee genetics, but when queens mate with a drone, um, they produce worker offspring by the queen fertilizing the egg with the sperm from that drone. Now, there happens to be this one gene that determines sex, right? We have the XY chromosome thing, and that's very different from the ways bees do it. They actually have a single gene, and that gene has lots of different forms. And so when a queen mates with a drone that has a different form of this gene, then all of the worker offspring that she fertilizes the eggs, they develop normally. Well, just by bad luck, by chance, sometimes a queen can mate with a drone that happens to have one of the forms of the gene that she has. And so what happens is half the time when she fertilizes an egg with the sperm from that drone, that individual has two different copies of the same allele, the same sex gene. And rather than developing as female workers, they develop into males. Diploid drones is the term you've probably heard of. And the workers go through and they cannibalize that because he's like a Franken drone. He's not going to develop normally. And so he's kind of just a genetic dead end kind of thing. So the queen's going along, laying all the eggs, fertilizing the eggs, has a good brood pattern. But 3 days later, you get all these empty holes after cannibalizing all the diploid drones. So the brood pattern is 50% and it looks like crap. So this is something that the queen can't control, right? She doesn't mate again. It was just bad sheer luck that she mated with, a dud drone. And so getting rid of the queen, and getting a new one in that lays more of a brood pattern like this is the way to solve that issue. Okay. So there is truth in shot brood being a symptom of a bad queen and, in fact, a poorly mated queen. But there's other reasons why you can have bad brood patterns. Here's a bad brood pattern, but you need to look a little bit closer and say, well, is that really a bad brood pattern because of the queen? If you look, you see some perforated cappings, sunken cappings, a hint of a scale, right? This is a bad brood pattern because of American foul brood, not because of the queen, right? Or something like this, right? This is a bad brood pattern, but you see all of these uncapped brown looking things. And this is EFB, right? This is a European foul brood. I don't think you guys really get chalk brood out your way, but that's another way to have bad brood patterns. And then, of course, the quintessential, where a lot of people blame their queens for this, which is parasitic mite syndrome, right? Where there's just the accumulation of parasitism by the mites, um, and vectoring all the different viruses that makes the bees sick, including the brood. You can get them pulling out all the brood, and you get this horrible brood pattern. But that's just because they're trying to keep on top of the mites. So, you can get brood pattern for bad brood pattern for lots of different reasons, um, not just because of the queen. So you really need to be introspective, when you're looking at the brood pattern. Now, we did this study with Katie Lee when she was a PhD student in Marla Spivak's lab in Minnesota. And we wanted to get to the bottom of, well, what's causing these bad queens? And so Katie's out there working with all these commercial guys in the upper Midwest, and every time she'd come across a bad queen that was diagnosed as having a bad brood pattern, okay, she would sample that queen, sample some workers, sample some wax, and she sent all of that to us. And then we measured the queens for their sperm counts and their body size and that kind of stuff. We measured the workers for different diseases and viruses. We measured the wax for like 200 different pesticides in them. All right. But then to have a comparison, she went to the colony next door that had a good queen, defined as having a good brood pattern, and she'd sample all of them and send those to us, and then we'd, be able to compare. And so in the first year when we did it, we had, several dozen pairs of these queens, and in one case it looked like, might have been a bad brood viability, another, or sorry, sperm viability of the queen. Another one, well, it might have been this one particular pesticide. Another one, it might have been this one virus, but there was nothing consistent. There wasn't anything across all the different queens, and whether they're good or bad, that was really there was no smoking gun, right? There was no obvious one factor that was standing out. And so we were kind of disheveled by that. And Katie came up with this great idea. And rather than just repeating the experiment, well, she said, well, why don't I take the bad queen and put her in the good colony and take the good queen and put her in the bad colony and then go back 3 weeks later. And if the bad brood pattern is really because of the queen, then this colony's brood pattern should get worse and this colony's brood pattern should get better, right? And that did happen sometimes, but most of the time it did not. It did not rescue, putting a good queen in a bad colony did not rescue its brood pattern, and then putting the bad queen in the good colony didn't make it markedly worse. So, the brood pattern is not really a great proxy for how good the queen is. It's how good the colony is. And if you think about it, so stop to think about it. The queen lays the eggs, but who raises the brood? The workers, right? So the queen could be doing her job. It's just that the workers aren't able to do their job. So really, the brood pattern is a symptom of the colony, not the queen. So outside of the times when the queen just unluckily mates with the wrong drone, most of the other time the brood pattern is something. It's a colony level trait, not a queen level trait. So keep that in mind. And you really, once you see a bad brood pattern, don't immediately blame the queen. Rule out every other reason why you could have a bad brood pattern before you blame the queen. That's my take home for that. All right. So now, again, it's not just the pattern that the queen's laying, but it's also what she's laying. And we know that as queens run out of live sperm in their spermatheca, well, she thinks she's laying an egg and fertilizing it, but if she's kind of shooting blanks, that egg doesn't get fertilized and then it becomes a drone, right? Because unfertilized eggs become drones. So if you see something like, well, this is different. Okay, let me back up. This is a queen problem, but a very, very different queen problem, right? For those of you that are studying for your master beekeeper exams or whatever, when you see multiple eggs in the cell, what is that an indication of? It's an indication of having a queen problem in the sense that you haven't had a queen in weeks. The workers have developed their ovaries and now they're laying drone eggs, right? So that's kind of like a hopeless queen problem where you can't really even requeen these colonies because once they get a taste of anarchy, it's pretty much impossible to bring them back to, the light. But what I'm talking about is something more like this. If you see these raised up drone cells in the middle of the brood nest where the workers should be, and it should all be flush, that's an indication, that the queen is starting to run out of sperm, live sperm, um, or that she has a lot of dead sperm in the spermatheca. Okay, so that again, that's that spherical organ in her abdomen that she releases some sperm every time she lays an egg to fertilize. And if she's kind of run out of sperm or the sperm in the spermatheca has died for some reason, then she can't fertilize those eggs, and you get an increasing frequency of drone brood where the worker brood should be. Now, that's very different from a pattern like this, which is all drone brood all the time from a young queen. Okay. That is most likely where she has no sperm. She's empty. Her spermatheca is empty because she failed to go on her mating flights or something like that, right? This can happen in Queens, especially in rainy weather. This time of year, out your way, you can have those systems come through for, a week or so at a time, and queens just can't get out to mate. But pretty soon, they still start need to lay eggs, and if they can't fertilize those eggs, it all turns into drones. So you need to remember kind of how old the queen is, and really looking at the pattern to read the brood nest and see not just how good the pattern is, but what she is laying. Oh, yeah, I forgot I put this in here. Okay, real quick aside. This whole idea that a drone layer, a queen that has either never got filled with sperm or is running out of live sperm, well, she's never going to mate again, so you just need to replace that queen. Well, we were doing this experiment where we actually wanted drone layers for one reason or another. So we were asking all our commercial beekeeper friends, if you ever see any drone layers, we'll buy them from you, you know. And so they did, they shipped them to us. And indeed, 40% of the queens that we received, they were indeed drone layers. But interestingly, 20% of them, they started laying drones, but then within a week or so, they kind of recovered and they started laying perfectly normal work. And then in another 40%, when we installed them into the colonies, they were not drone layers at all. So what that suggests to us is that drone laying might actually be a temporary thing where the sperm might not always be dead. But maybe there's a kink in her reproductive tract where she can't release the sperm or something like that, and just putting her in a cage and shipping her to us through the mail kind of worked that out, whatever that is, and now she can start fertilizing eggs again. So there may be a way to remediate these kind of drone laying queens if you're squeamish of trying to go out and replacing her. But more work needs to be done on this. I just always found it interesting that it's not necessarily kind of a terminal condition for queens. All
A
right,
couple. Oh, can you
go
hear
ahead.
me? There we go. We have a couple of questions in the chat. Or actually, it looks like one. So there was, let's see, Greg is asking if swapping the queens at 3 weeks doesn't change the shotgun egg laying pattern, and it is related to the colony rather than the queen. Um, then what happens 6 to 8 weeks later when the nurse bees are replaced by the new queen? Does the brood pattern change?
Yeah, great question. And that's actually one that comes up, um, where what if we just allowed this colony to go longer, or this experiment to go longer? Um, don't know. So you're right. Once a brood pattern and kind of the demographics within the comb is fixed, right? It might kind of take a while for them to work that out. So it's possible that we might see something different giving a longer period of time. But that would be a whole nother experiment or kind of double the same experiment. So yeah, so we're not really sure. But it's still not immediate, right? It's still, I think, more a symptom of the or of trait of the colony more than it is of the queen per se. Good
Got it.
question.
Thank you.
All right, so let's get back through all of this. Um, I'm just gonna I'm gonna skip some of this stuff. I want to talk about reading the comb. I love this picture. I got it off the internet. Whoever took it is probably my favorite person ever because it really shows how you need to read the comb when it comes to queen cells and where you're at in the requeening process. Okay. Colonies under swarming or supersedure, they'll raise a couple dozen, queen cells, and they can be cryptic. They can be hidden. They can be off on the side. They're not always obvious. And they can raise them in bunches. But what happens is that the first queen, she will chew her way out kind of from the capping. So you have this perfect circle on the bottom of the cell where she chews her way out and pulls herself out. But virgin queens are very reluctant to share the throne. So they have to dispatch of all of their sister rivals. And so whenever they seek out and they find other queen cells, what they do is they chew a hole in the side and then sting the pupa of the developing queen, which then kills them. The workers come along and say, "Hey, what's this dead queen doing in this cell?" They chew the hole out more and then they get rid of the dead queen. So this is a queen cell where she emerged and then she went and immediately assassinated all of her sisters right next door. Right? So you know that there's a queen running around that colony, and it can be really, really hard to find a virgin queen because they have the abdomen size of a worker. They don't really stick out very well, and they're very fast, and they like to hide, and it can be really hard. So if you try to throw in a mated queen in a cage into a colony like this, you're just waiting for her to get killed by the virgin queen too. It's almost better just put everything back, let that virgin queen kill everybody else off, go out and mate, come back and start laying eggs. Okay. This is a good sign of when that virgin queen is about to come online. They really start to polish the middle of that brood nest. They don't have any more brood because whatever brood was left from the previous queen, that's all been hatched out by now. And so the workers are just eager and just ready to go. So if you see this kind of beautiful polished nursery right in the middle of the brood nest, you know that she's about to start laying eggs. So anyway, but we're here talking about queen longevity and how long queens live. And we did this experiment again where we had high quality queens and low quality queens. And one thing that we were predicting is that the low quality queens were so bad that the workers were going to recognize this and replace them because they were just so bad. But that actually didn't happen. These low quality queens were not replaced more frequently than the high quality queens. In other words, it did not induce supersedure, and therefore lower queen longevity. So we really didn't know why, but the workers don't seem to be queuing in on the same quality measures that we queue in on as beekeepers. And so that started me down this rabbit hole, and it caused me to develop this kind of conceptual model. And this is really complicated, but I'm going to walk through it, and it's going to make total intuitive sense to you, I promise. If you look at the queen's lifespan, you just look at it over time, and this is not linear, but you look at it over time, you can really break down the queen's lifespan into four different phases. All right. The first phase is the development phase where they're actually being reared, and then they emerge, and then they mate, and then they start laying eggs. Okay. That takes about 3 weeks or so. So that's the development phase. Once she starts laying eggs, then that's the establishment phase, and all the workers in the colony are from the previous queen, either her mother or whatever other queen, that because we're often shipping queens and inserting them into new colonies and things like that, right? So there's handling that we do at the boundary between these two phases, right? But once the queen starts laying eggs, it takes about 9 to 12 weeks for all the old workers to die off and the population turns over so that it's entirely her offspring. And that's the end of the establishment phase. Okay. The third phase then is the survival phase. And this is what we want to maximize. We want this to be, 3, 4, 5 years, where the queen just kind of keeps doing her thing as long as possible. But at some point, queens are not, they don't live forever. So even the longest lived queens need to be replaced and they eventually fail. So that final replacement phase is where the colony decides off with her head, make a new queen, and then the cycle starts all over again. Okay? So four different phases of the queen's life. Now, the number one thing that really kind of dictates how long a queen can live, well, yeah, as long as she has sperm, she can keep fulfilling her function, right? So in the development phase, when she's a virgin queen, obviously she has an empty spermatheca. When she mates, it becomes full. And then as she start laying eggs over time, she depletes the sperm in her spermatheca, fertilizing those eggs. Okay. And then once she runs out, she becomes a drone layer, and ideally she would be replaced. Okay. But, the workers are not like, they can't tell how much sperm she has in her spermatheca. They can't really assess that. They must be kind of judging her in some other way. And so, not really knowing what those things are, we can just conceptually say that, okay, there's some sort of gestalt perception of the worker and of perception of the queen by the workers for her uh, quality. And either she's acceptable, that they detect that she is good, or that she's unacceptable. And so there's some sort of threshold that has to be crossed that, the queen early on, okay, she's acceptable. And at some point, her opinion poll by the workers dips below this threshold. And now all of a sudden they're like, she's no good anymore. We need to get rid of her, right? It's kind of like us with elections, right? We really only have two choices. It's either keep the incumbent or throw the bum out, right? And it's the same thing with the workers. There's no halfway here. It's either you keep the queen or you make a new one. And so they have to have some sort of collective decision process of how this opinion poll is good one day and then not good the next. And my contention is that whatever is driving this collective decision making, this opinion of the queen, is being kind of pushed earlier and earlier into the queen's um, life cycle so that she's being requeened earlier in the survival phase, or in many cases, even during the establishment phase. We've done experiments. I've heard other beekeepers have their colonies requeen newly mated, perfectly good queens from their packages. But there's just something about the system where they just don't have a good opinion of that queen and they replace her only within a few weeks rather than months or years. Okay. So this is a very complex process of how a colony makes these decisions, right? How do the workers kind of sense that there's something wrong with the queen now where it wasn't before? And then it goes back to all of these factors that we talked about before that can interact with all of these different things within the colony in very complex ways that then results in this collective decision making of getting rid of the queen or not. And so colonies can make mistakes, right? They can get rid of a perfectly good queen, or they don't get rid of a bad queen. And so the more that we can understand kind of this psychology of the workers, I think that'll really tell us a lot about what's going on with the queens and the problems that they're experiencing. But it's very, very complex. And it's something that is going to take a lot more research to suss out. But in the meantime, this is where I stand, and this is how I think of it now. And hopefully I've convinced you to do the same. That, why do good queens go bad? Well, we know lots of reasons why. But in the end, is it the fault of the queen or is it the fault of the colony? And by and large, yes, there are some things that can result in bad queens, but most of the time it's the environment in which the queen is placed that really the colony is responsible for why these queens are having problems. And it's kind of like with us. Well, like if the economy isn't doing well, well, let's vote a new president in and see if we do any better that way, right? The workers are kind of doing the same thing. Well, we can't really do anything about these viruses, but maybe if we get rid of the queen, that'll improve the colony, right? So the way I really think of it now is that these queen problems are a symptom of other things going on in the colony, of other stressors that the colonies are experiencing. And they're kind of blaming the queen, and it's not actually the fault of the queen. So, if we can make sure that we keep our mites under control, if we can make sure that they have good nutrition at all different times of the year, I think those stressors will alleviate a lot of these queen problems, and that they don't kind of collectively decide to get rid of her in a kind of a, a desperate attempt to try to solve them. So, with that, hopefully you've enjoyed the little journey of my thinking and my thought process about these queen issues and kind of redefining how we look at them. And by doing that, that might improve our ability to manage them. Thank you very much.
Thank you, David. Thank you, thank you, thank you. We're all clapping, but I don't know if you can hear us. So this will be a great opportunity to ask some questions. Although this is what happens, David, is we all our brains are swimming and we're absorbing everything that you've presented, and then we don't have any questions. So,
Quite all right.
but hold on, let's see. I'm going to give everybody just a minute to see if there are any questions. Judy, do you have any questions? Yeah. Okay.
I really liked what you said there at the end, is kind of bringing the responsibility for a strong colony back to our management, in that there isn't anything else that the workers can do except replace their queen. And, whatever the situation, that's all they can do, or we can do other things. So.
Well, it's one lever that they can pull, right? And I think just kind of instinctively, that's one thing that they can do, and it's not necessarily a cure-all, right? But it's an option that they have. And again, the more that we can kind of study the psychology of the bees and what are they queuing in on, well, then we might be able to tweak that and prevent them from getting rid of perfectly good queens, or even induce them of getting rid of bad queens, right? So we might be able to work with the bees and have them make, perhaps more informed decisions rather than rash ones. So we're still kind of in the early stages here when it comes to this, but I really think that looking at the queen problems as a symptom rather than a cause will improve our beekeeping.
Good deal. Good deal. All right, I'm looking around our room because I want to make sure that our folks here that attended in person have the first opportunity for questions. So anybody with questions or thoughts? Anything else they want to add? See, our brains are all swimming.
Well, so I see a question here on the
Oh
chat.
yes. Do you want to
We're
go for
talking
that?
about the
Yeah.
latest article in Bee Culture that's highlighting supersedure issues tied with viral infections. Yes, that is our work. I have not incorporated that into I made up a whole nother talk on that. But going back to that kind of very messy Gordian, knot of all these interacting factors, we've started identifying some of these major ones. And one of those is viral infections of the queen. What happens is that tends to shrink their ovaries, which then means that they are not laying as many eggs. Now, it doesn't necessarily mean they have worse brood patterns. It just means they're not laying as many eggs. But again, the workers can't really tell this directly. But what happens, and what kind of that the research is really showing, is that it's the brood pheromone. So the queen is producing pheromones, right? But that's not the major signal that the workers use to say off with her head. The major signal is actually the pheromone that the larvae are producing. So if the queen's not laying as many eggs, there's not as much larvae in there. And the workers are like, hey, something's wrong with our queen because I don't smell as much brood. And that's one of the key ways that they kind of sense whether the queen needs to stay or go. So, viral infection, brood pheromone, these are the main kind of culprits of what's going on with this. Um, and I think we're starting to get a little bit closer to really getting some of those answers. But thank you for pointing out that bee culture article. I had forgotten that that was covered in there.
Yeah, good deal. Do we have anybody online with us that would like to come unmuted and maybe ask a question? Let's see. All right.
Maybe if you can hear me.
Oh, yeah, we can hear you. There you go.
Okay, cool. This is one of those non-scientific questions. All of the inputs that we're putting in, especially homemade sauces that involve thymals and all those kind of things, is there any interplay there with pheromone disruption? I mean, you open up a hive where somebody's put in a cake of thymol. They've used a bunch of different essential oils. That's loaded with all sorts of camphor smells. And you have any issue that you see there when somebody does it?
So the answer is we don't have complete data and knowledge on that. That's the short answer. The longer answers is that the brood pheromones are plural and they act in different ways. So there's kind of the young brood pheromone called E beta ocimene, and that's volatile, right? So that kind of gets out into the air and kind of it permeates throughout the nest, right? But then there's an older brood pheromone called the brood ester pheromone, which are kind of like oily secretions. So it's not volatile, it's passed around through body contact of the nurse bees. So, I would, if I were advising somebody to test that, I would say, well, you might want to look at how thymol or some of these other fumigant, acaricides might affect. I would predict that it might affect the ocimene signal more than it would the older brood pheromones, right? So it can start to get complex with that as well. But the bees have such an amazing sense of smell. They can walk and chew gum at the same time with different smells. So I could see them overcoming it as well. So we just don't really know. I do know that one of the major ways that you can really start to see induced supersedures when you try to impose a brood break in the middle of the summer, or if you at least here we use a lot of formic acid in the spring, which can burn a lot of the young brood. Well, then you start getting supersedures. Why? Because there's no brood in there, right? And the workers are, well, we're not smelling the same amount of brood pheromone that we should, and that can lead to that. So there's no guarantees. This is all probabilistic, but it's very possible that those types of things could have an impact, but we just need to connect some more of the dots. And this is also, Mark, where it's really, really helpful for, scientists to be interacting with the beekeepers that are doing these things, because anecdotally, you do this to 200 colonies and you're like, geez, half of them they superseded. Oh, okay. That's something that is worth looking into more. But if it's something subtle, it's something that we can all miss, right? And so I think trying to to think through it together is really, really helpful. So thank you for that comment.
Thanks.
All right, very good. All right, last call for any questions for David. All right. Going once, going twice. All right, I think we're good. David, thank you for staying up late with us again. We really appreciated your time and knowledge tremendously.
Thanks so much, guys. It was great to see you again.
Nice.
You have a good evening.
All right, take care. All right. So let's see. I'm just going to stop our recording here.